Okay, so today I'm going to talk about the update of the analytical technology in the biological development. Oop, okay, so as you know, the biology is a rapidly evolving field, so there's a need for the analyzing the complex biologic. So I have three topics. Now I introduce the biologic manufacturing and bioprocessing. So this is a very typical slide. Before you do the manufacturing, you need to do several years of homework, discovery the target, and then you develop your antibody. You have to make your so-called cell bank, right? So cell bank is a critical reagent. That's the first thing. And then you sort the vial to do the upstream, downstream, and also analytical to support all the manufacture process. Okay. Manufacture is a simple thing. There are three components, very important. Number one is the raw material. You make anything, you need raw material. The raw material need to be qualified. And then secondly, your process. You need to identify a process parameter. You should have some sort of range. And then you do a GMP manufacturing, the control. Okay. So that's a biologic, the monoclonal antibody. For the AAV, again, you have to do some homework before you manufacture. So you have the viral gene into the vector. You have the other AAV helper and the helper. Three things is called triple transfection into the 293 cells, and then you have to do manufacturing. And this is a very quick summary of the AAV. At high level, you need to make a cell bank. The cell bank means plasmid DNA, which is E. coli, and you make a GMP material for the plasmid, and then you have to have a qualified or the 293 to the downstream, upstream and downstream, and analytical. So basically, even it's a gene therapy, the overall process is very similar: downstream, upstream, downstream, and analytical. Okay, so again. As everybody knows, FDA published 20 years ago about the PAT, and PAT has been utilized in small molecule for a long time. It's very successful for the biotech product. It's going there. Again, it's making a lot of progress, but for the so-called gene therapy, there's a very slow progress. So BioForum, as you know, there's a UK and global, consortium from every big or small biotech together. They published a white paper 2 years ago, 3 years ago, and then most recently published another paper. So this is a summary. They think when you develop the PAT, you need to consider viability, cell growth, and also titer, etc. Okay, so in terms of the monitoring, you need to have 4 different kind of monitoring. Ideally, you should have inline, online, but sometimes a lot of company or CDMO, they do offline and inline. Okay, so once you have the process, you put into a digitalized process, combine together, and become a so-called digital twin. This is very general idea of the manufacture process. Okay, now we go to the second part, which is the quality attribute, right? So for the any biologic, the quality attribute become 2 sections. One is called analytical and the QC, which is always under the GMP guidance. But the other part is called characterization, which is not really GMP, but still very important part for filer IND or BLA. So on the left side, you have 7. You have a safety, identity. I'm not going to read through all of this, but basically, your quality attribute including the following thing. And this is a very summary slide based on ICH Q6B. It's, every drug substance, you have 7 different quality attribute category. The most important thing I highlight in red color is impurity and purity. That's the most FDA. They look at the purity and impurity very seriously. That's number 1. And number 2 is the potency. And other thing also important, but these 2 things is most important thing. And then in terms of drug product, they emphasize 2 things. Number 1 is particle, including subvisible particle or visible particle. The other one is make sure it's sterile because you are going to inject into patient, right? It has to be 100% no bacteria, no yeast, nothing else. Okay, I have following 3 slides just to summarize in terms of the quality attribute. The antibody most has a so-called post-translational modification. So to do that, you need analytical technology, right? And then when you talk about common PTM, you have the glycan structure, which is very complicated. You have amino acid, chemical degradation. It also needs analytical technology, okay? And then the third thing is fragmentation, aggregation, and also charge variant. I'm highlighting in color, which is the thing we are going to talk about today. It's also require analytical technology. So that's a high level. Understand, when you do all of this, you require technology. In terms of the gene therapy, again, you want to have, ideally, you have the so-called purity, right? On the left-hand side, if you have a full capsid, that's good. But in reality, you got a lot of partial fill, you have the empty, and you also get aggregation. And all these 3 things, you need to document it and make sure you can minimize as space you can. And then in terms of the potency, there are 3 different potency. Number 1 is infectious. Number 2 is a genomic titer. And number 3, capsid. There's a high level. This is a quality attribute, right? Again, we always focus on 2 things. Number 1 is impurity, and number 2 is called potency. Impurity also including a contaminant, which is very important as a safety issue. Endotoxin, bioburden, mycoplasma, and adventitious virus. And this, again, a very similar slide, just show you, all this bioactivity is based on the genomic titer, infectious titer, and capsid titer. Okay, we come to the main part of this talk. We want to update the analytical technology and the trend in a biological manufacturing. So why we need the new analytical technology? Because emerging therapy, when you get into a cell therapy or gene therapy, you have new modality. So you need an emerging therapy, characterization to characterize all this complex biologic. And the bottom line is to apply analytical technology to bioprocess, number 1, and number 2 is to manufacture to the final drug substance and drug product. Okay, this is a very important slide, just to give you an idea. The current analytical technology, I basically break down into a category. Okay, you have so many different assays, right? But the category is number 1, chromatography. That's the most important, powerful thing, especially for a small molecule, right? Number 2 is electrophoresis. And number 3 is mass spec. And then the following, I'm not going to read through. All of these are important for characterize the complex, biologic. Today, we are going to focus on this new innovation, automation to make faster of the assay. We're talking about spectrophotometry, about, VPE, Raman spectra, and also focus on the glycan structure, ICMS. All this we're going to keep it over high level. What's the trend and what is the current future is going to be. Okay, so in terms of PAT, that's based on the ICS 20 years ago. They have design, analyze, and control. So basically, if you look at the upstream on the right side, you have to monitor, your DO, your oxygen, your pH, everything. And downstream, the same thing. You have to monitor all of these things. This is a high-level summary. And then this is the breakdown. When you do the upstream or downstream, what is the sensor right now? So basically, right now, most of CDMO, they still use the individual sample sensor on the left side. pH, you have the sensor. DO has the sensor. Dissolved carbon dioxide, glucose has sensor, lactase has sensor. They have different sensor. But the current trend is going to be kind of, combined together into one sensor. So that, the lactase sensor, lactose, the glucose sensor, they are individual, but you may be able to combine together. So that's the thing. Recently, you can see Repligen, data booth over there. Repligen, they bought a 908 device, the Maven online. They can, monitor glucose and lactate in one probe. It's a two probe together, but it's one probe. It can do the align or inline analysis. So at the end of my talk, I will talk about Raman spectral. This is a future trend, and I know a lot of companies, especially big pharma, they develop this one, kind of to consolidate all these different, sensor into a few or maybe one. That's the future. This is not a new technology. It's introduced about more than 10 years ago. It's called solar VPE. And most of the CDMO right now, they use this to do the assay. So it's based on a slope. So you basically have a variance, and then based on slope, you can determine a concentration. And this another one, you want to do the in-process monitoring. Instead of take a sample out and then do the offline assay, you can do the inline assay. It's called flow PVE. Again, this is developed by C Technology, but it's bought by Repligen right now. So this is the trend. I know a lot of big pharma, they use the flow PVE to do the inline assay, especially in the final UFDF drug substance manufacturing. They want to know the concentration instead of taking a sample and do the offline assay. Okay, so this got to the point is the Raman spectral. So Raman has been used in the raw material identification for a long time. A lot of raw material are small molecule, right? They have a very distinct, very fingerprint, very distinct the Raman spectrum. So you can use the high ID. So that's very traditional way. But nowadays, people start thinking about use this one to do the protein ID. Okay. So as you know, protein ID, you can do the, ELISA, you can do mass spec, you can do, different assay to get the ID. But the ideal case is that you can do, use the Raman, just do the ID. For example, if you have the fake biologic in the market, you want to know immediately, instead of take a sample and do assay, take a long time to get a result, right? So Raman become a trend to do the ID test. And this is the paper published in academic in Ireland, about 2 or 3 years ago. And then they give a case study. They use the Raman analyzer, and then they based on the 3 different antibody, they figure out what's the fingerprint of 3 different ones, and they can identify which one is which. So that's for identification of the protein drug. So has been used in industry too, but this is a trend going to do the protein identification just by a simple Raman spectroscopy. Okay, and this is the slide I'm going to present because you can see in this conference or this Bio 2026, the Future Film, they had a booth over there. Taiwan Biomanufacturing Corporation, they had a booth over there. Both of these two, they have the demonstration to use the inline process monitor by Raman spectral. So it become an industry trend, people trying to use one Raman spectral and then to identify maybe lactose, glucose, and some metabolite at one time. So that's the future trend. So if you're interested, you can visit this too. I think Future Film, Koriba is a very good one. You can talk to them. They can explain to you, how this work. They don't show data anyway, but they told me, they can do a lot of, metabolite in one probe. Okay, this is another trend because N-glycan analysis is very important, right, for the molecular antibodies. So nowadays the trend is to the automation. As you know, when you do glycan analysis, you have to use enzyme to create glycan, and then to fluorescence, and then to the, heated column, a lot of procedure. A lot of things can happen in between procedures. So they are trying to automation and to get the streamline, result very quickly. And two company, Waters and Adrian, they do have a kit. So you can buy their kit, and then you can develop your own to make a automation, make sure your data is reproducible every time. Okay, this is another trend. It's called rapid microbiology method, RMM. because cell therapy, especially cell therapy, you do the sterility test, it takes 14 days. And when you manufacture the material, you want to get a patient, in no time, right? So you want to get it rid out as early as possible to make sure it's safe. There's no bacteria, there's no yeast contamination. So the technology is there. This is all, not all, the most recent. You can base on viability or based on PCR, whatever, And actually, Lonza just bought the red one last year and close the deal this year. And they basically use the viability-based, assay. So this is a relatively new. However, it just came out about 3 months ago. A French company is called Bio. They introduced a new, based on AI model, a new system called Louis system. It's based on the so-called transformer multimodular spectrum. Basically, they have so many different bacteria. They shine a light to do the UV visible, all the spectrum, and they identify which spectrum belong to which microorganism. And then due to AI, machine learning, they introduced the product 3 months ago, and they claim they have validated some of the results. So this is a new thing. They don't need to do the viability or growth, anything. They just take a sample and then get a result. So this is something we need to watch out in a future development. So this is the trend of the so-called rapid microbiological method. Again, ICF is very important for the charge variant, right? Because monoclonal antibody, they have different charge. And FDA at the end, when you do a BLA, they want to know which residue, which fraction is acidic, and what kind of structure there. So there's 2 companies. One is called Protein Simple. They introduced Maurice about maybe 6 years ago. And then you can collect a sample to do the mass spec. This is offline analysis. However, again, this is an example. They use a map, use gene therapy, AAV. They demonstrate they can do this. However, the most recent, about 3 years ago, SciEx, they bought Antebio, the small company. They do the ICIF. They link to their mass spec and then to do the inline characterization of the monoclonal antibody structure: acidic, basic, and the main peak. So this is the thing you should be aware of. And then we go to the HCP impurity, right? HCP, as everybody knows, in the old time, it's ELISA. Most recently, a lot of companies moved into the mass spec, although they still use ELISA. And the USP published ELISA in USP 1132. It's called ELISA. And then they published another one about, I think it's two years ago. It's 11.1, which is the best mass spec. So USP recognized by mass spec to enter the HCP is very powerful and become a trend to do the HCP, HCP identification and quantitation. Okay, next thing, in terms of mass spec, is called multi-attribute method, and it's introduced by Amgen about 10 years ago, and then it became a trend. Amgen filed a lot of IND and even DLA. They try to use the MMN to do the QC release. Although FDA said you have to do a lot of work in terms of compare the traditional method and the new method. Okay, I will talk about that. So basically, MMN is not a new thing. It's a peptide mapping, which is reference standard. Period. Every time you run peptide mapping, you have reference standard, and every lot of the material you compare that, you can see whether it's different or the same. Is any new peak, new impurity come out? You can analyze by HPLC very quickly. So that's a workflow. And then MMN is trying to replace the conventional method. It's not there yet, but it's going there. So basically, MMN, they can get a lot of information. They can get the so-called charge. You can see charge and also get the fragmentation. You can get glycan structure, especially glycan structure is very powerful. But one thing they cannot do, is aggregation. So you still have to do side exclusion to do aggregation. Other than that, they can replace all this method. Okay. So now the trend is to apply this technology, even though it's not very new, gradually going there to replace the QC and stability testing. And again, the USP chapter 1060 is published there. They already think MM is the future to do the mass spec, QC, and also stability testing. Okay, so you have MM, right? Use a mass spec. Now Genentech come up with another idea. How about we use the Raman to do the multiple attribute? In this case, it's Raman spectra. So basically, they think they can use one instrument, Raman, and then to analyze a drug product. As you know, drug product has a formulation, right? Has a protein, number one. Has a buffer. They have a surfactant, right? And they have a stabilizer, sucrose. They have a methionine. They do all of this. They think one assay, they can get all these 5 in one assay. It's called multi-attribute assay. It's published about 3 years ago. And then you can see all this workflow. Basically, you analyze individually, component in a Raman, and you put together and then come up with a fingerprint or a signature, and then you can get the product quality attribute. And they do a case study. Yes, they can do. However, they can do the protein concentration. That's good, but they cannot do buffer. They cannot do the surfactant. They fail validation. They can do osmolarity and they can methionine. Okay, so this is another trend. I don't know how it's going to be. Use a Raman to do the multiple attribute method. Okay. And then we come to the gene therapy, right? Gene therapy, the main thing is empty, full, and empty and full. So 4 years ago, there's a draft guidance for FDA to consider. That's from consultant. They can, based on the current, technology, they write a paper to recommend, for the AAV critical quality attribute. What should we do method and what we should do. So basically, let me get a little background. The full and the empty traditional based on the gold standard is called AUC. So basically, you take your sample, you spin for a day, and then heavier one go down, right? And the empty one is on the top, so you can do the distribution. So that's very low throughput. You have to do one sample. Basically, I did that before. It about most of one spin is about 4 samples, and it's very very low throughput. So basically, you can do another technology. They recommend 5 different technology. Okay, TEM is another one, but TEM is based on how you slide your sample, and every time you might get a different variation of your result. So the traditional one is AUC, gold standard. Okay, and then recently developed a new technology called CDMS. So this is another white paper published there. They summarize all these 5 different technology. And the most promising I highlight over there. Number one is CDMS, and the other one is AUC. Again, it's a gold standard. And other things still okay, but it's not really, recommended by this, white paper to FDA. Okay, so we come to the last one. Is the CDMS. Okay, so CDMS is based on the charge detection mass spectral. This is also a mass spectral, but it's for the high molecular weight molecule. This technology, basically, they can measure the M over charge, so mass over charge. At the same time, they can measure the individual particle, the charge. So mass spec is based on mass per charge, right? If you know the charge, you can do the mass immediately. So they get a very, precise or accurate mass for the big, molecule. AV is very big, right? So they can do that. So water come to this. Let me go back a little bit. So this technology is developed by Indiana University. There are 2 professors. They form a small company. It's called Mega Dalton Solution, and then Water basically bought the company, buy their, bought their patent, and then develop the instrument. And as far as I saw, Water's presentation is quite powerful because you can do the mass spec, you can plot the M over here on the x-axis and the charge on the y-axis. And based on that, you can calculate the molecular weight. The beauty is that they can do the quantitation of the, the full and the empty. So you can see on the right side, they have the full, empty, they have the empty 100%, empty 0%. They have the 5 different calibration curve, and they use their instrument. They can do the linearity, get R square. It's quite good. So I think this is a trend in terms of AAV to analyze the empty versus full or partial ratio. You can do this more convincingly, more accurate instead of use AUC. I think that's my last slide, and I welcome all the questions. Just give you the whole idea. What is the current, technology using all this biologic, including monoclonal antibody and also gene therapy. Thank you.
Lovely, thank you, Kevin. Does anyone have any questions?
Yeah, thank you. Nice talk. So I have a question regarding the AV, like an empty-full ratio, because it's rare to define full capsid, right? Because when you are produce the AV, it could be partially packaged. So empty is very absolute, right? You can easily tell, okay, it's empty, it's empty. But for full, how do you really define full? I mean, if you cannot fully define full, how do you define the empty-full ratio? So another question is that, like for the still the characterization of the full capsid, like can you comment on the NGS technology for the full capsid characterization? Thank you.
Okay, so the first question is how do you know the AAV is full, right? That's the first question. Yeah, it's full. So basically, when they run this experiment, they have they had a full basic. I remember, I think USP. I'm trying to look at yesterday, but I didn't look into that. The USP has the full AAV standard. I think yes, right? I remember they just recently released a standard because USP, has to provide a standard, for a customer to have a kind of standardized AAV, If it's already in the market, I think I heard they put into it's already in the market, right? So they want, it can prove it's full. The second question you ask is about the NGS, right? So NGS, you basically look at the sequence, right? So I don't think NGS is quantitative enough. I think in the old way, they use the qPCR, in the old way, qCPCR to determine the genome copy number. That's number one, is DNA level. And number two, you want to know the capsid, right? And capsid, again, use ELISA. So there are two different methodology, and let's try to correlate, the DNA and also protein, which is the capsid, together, yeah.
Yeah, thank you. Yeah, I do have a follow-up question. So regarding the like qPCR, ddPCR, right? So because I was working on AAV production before.
Okay.
Yeah. So when I was running the qPCR as well as the ddPCR, I always see a kind of consistent difference between qPCR data and the ddPCR data. I don't recall which one is higher, but it's very consistent across different cell types. So my question is how much difference is tolerated for the manufacturer standard? Like a variation, for example, like sometimes I see like I said, I don't recall which one is higher, like 2 times higher regarding the VG titer.
I think this is a hard question because all the data is coming from 2 different methods, right?
Yeah.
And 2 different methods based on 2 different methodology. To my knowledge, the FDA, they really look at your methodology and it's no way or difficult to compare 2 different results.
Yeah.
That's my opinion.
Yeah.
Yeah. I think that's a tough question, and reproducibility is very important.
Yeah.
So if you have, again, you have to analyze, the so-called standard, right? And then you kind of correlate with the standard to come up with your quantitation result. That's my answer. I think the answer might not be very pretty, but basically two different methods always get different proposal anyway, right? As you know.
Thank you so much.
Thank you.
Do we have any other questions? In that case, can we please thank Kevin one more time?
Thank you. Thank you very much.